Good morning students. Today we are going to learn about the topic name is lipoproteins. Introduction. Lipoproteins are molecular complexes that consist of lipids and proteins. The protein part of lipoprotein is called as the apolipoprotein. It is usually abbreviated as LP. That means lipoproteins are usually abbreviated as LP. They function as transport vehicles for lipids in blood plasma. Lipoproteins deliver the lipid components such as cholesterol, tag means triacylglycerols to tissues for utilization. When we come into the structure of lipoproteins consist of inner core. It is hydrophobic in nature. It contains hydrophobic triacylglycerol, cholesterol ester and tails of phospholipids. And this inner core is surrounded by a coat shell of phospholipids, apoproteins and cholesterol. The polar head portions hydrophilic of phospholipids and cholesterol are exposed on the surface of lipoproteins so that lipoprotein is soluble in aqueous solution. That means the polar head portions are hydrophilic in nature so that lipoprotein is soluble in aqueous solution. The apoproteins also increase the solubility of lipids. That means the protein part of the lipoproteins also increase the solubility of lipids. And then next, structural organization of lipoprotein. Then we see the apolipoprotein and cholesterol and phospholipids are present on the outer side, and triacylglycerols and cholesterol esters are present in the inner core. The inner core is hydrophobic, whereas the outer surface of the apolipoprotein and cholesterol and the phospholipids of the lipoprotein is hydrophilic in nature. When we come into the classification of lipoproteins, depending on the density by ultracentrifugation or electrophoretic mobility, the lipoproteins are classified as 5 major types. First one, chylomicrons. It contains apoprotein B48. And the next one, very low density lipoproteins, simply called as VLDL, or it is also called as pre-beta lipoproteins. These VLDL contain main apoprotein is B100. And the next, intermediate density lipoproteins, simply called as IDL, or also called as broad beta lipoproteins, And the next one is low density lipoproteins, simply called as LDL. LDL are also called as the beta lipoproteins, and this major lipoprotein present in the LDL is B100. And the next one, high density lipoproteins, simply called as HDL, are also called as alpha lipoproteins. Major lipoprotein present in the HDL is ApoA. And the next one, free fatty acids or non-esterified fatty acids are complexed with albumin. Albumin is a protein. When we coming to the chylomicrons are synthesized in the liver and transport exogenous dietary triacylglycerols to various tissues. This is the function of the chylomicrons. Those are the synthesized in intestine. It contains the apoprotein B48 and they consist of highest quantity of lipid and lowest concentration of protein. That means chylomicrons contain 99% of lipid and 1% of protein. The chylomicrons are the least in density and largest in size. The next one, very low density lipoproteins or VLDL. These are also called as pre-beta lipoproteins. The main apoprotein present in the VLDL is B100. They are produced in liver and intestine. And VLDL are responsible for the transport of endogenously synthesized triacylglycerols. The next one, low density lipoproteins, LDL. Those are also called as beta lipoprotein. They are formed from VLDL in the blood circulation. They transport cholesterol from liver to the other tissues. And the next one, high density lipoproteins, HDL, also called as alpha lipoproteins. Major apoprotein in HDL is ApoA. They are mostly synthesized in the liver. Three different fractions of HDL are identified by the ultracentrifugation process. Those are HDL1, HDL2 and HDL3. And the next, HDL particles transport cholesterol from peripheral tissues to liver. That means reverse cholesterol transport takes place by HDL. When we coming to the apolipoproteins, the protein part of lipoprotein is called as apolipoprotein, simply called as ApoLP, or also called as apoprotein. All apoproteins are synthesized in liver. Small quantities are produced from almost all organs. Intestinal cells produce small quantities of ApoA. They will solubilize the lipid part. When we coming to the schematic representation of ultracentrifugation, electrophoretic mobility of lipoproteins, in ultracentrifugation, the lipids are separated based on the density. By the basis of density, the lipoproteins are separated in ultracentrifugation. In this, high lipid content will have low density. That means chylomicron have high lipid content and low protein concentration, because it having low, that's why it having low density. And the free fatty acid albumin contain high density and low lipid content. And when coming to the electrophoresis, electrophoretic separation is based on the mobility. That means cellulose acetate is used for this process, and oil red O dye is used in lipoproteins mobility by electrophoresis. In this high protein content, lipoproteins will move faster. That means lipoproteins having high protein content will move faster. And this diagrammatic representation, chylomicron, LDL, IDL, VLDL, HDL and free fatty acid albumin is seen. Next one, functions of specific lipoproteins. First one, chylomicron carries dietary lipids from intestine to liver and other peripheral tissues. And next one, VLDL carries triacylglycerol and cholesterol from liver to peripheral tissues. And then next, LDL, that means low density lipoprotein, receives cholesterol from VLDL and HDL and carries it to the peripheral tissues. And the next one, HDL receives cholesterol from peripheral tissue and return back to the liver for biliary excretion. When we coming to the function of apolipoproteins, and the first one is the ApoA1. It activates LCA transferase enzyme and is also acts as ligand for HDL receptor. It is a anti-atherogenic and it is specific for HDL. And the next one, ApoB100, only apoprotein component present in the HDL. That means the ApoB100 protein is present only in LDL. It binds to LDL receptors on tissues. It is one of the biggest proteins having 4536 amino acids and molecular weight is 550 kilodaltons and it is synthesized in liver. And the next one, ApoB48. It is a major apoprotein of chylomicrons. It is synthesized only in intestinal cells. ApoB100 and Apo48 are products of same gene. Those are the products of same chain. In intestine, mRNA undergoes editing to produce only B48 protein. B48 is named because it is only 40% of the size of B100. And the next, ApoC2. It activates lipoprotein lipase. And the next one, ApoE. It is a arginine rich protein. Thank you.